All right. So, well, good afternoon, everybody. I'm going to paraphrase Antonio and say that I'm sure you're all super excited to hear about taxonomy, right? Well, okay. Well, I want to thank Simon for inviting me to give this talk. I think it's a little bit off topic, but I hope to make it interesting. You already saw that taxonomy can be interesting from Arvind's talk, the way he named viruses. So we try to make it interesting and fun. And let me just say, I'm based in Brazil. I'm currently here on sabbatical, hosted by Simon. And I'm also the president of the ICTV, the International Committee. So I have all these different jobs right now. And I'm going to start by explaining why do we do taxonomy, right? I mean, many of you are aware of that. Many of you actually are involved in taxonomy, but maybe many of you are not. And that's basically the reason why we do taxonomy, okay? Makes our life easier, all right? When it's well done. Bad taxonomy actually can kill you, as people who have eaten poisonous mushrooms instead of the funny ones will attest, or maybe they will not attest. So that's why we do. Okay, and I don't want to spend too much time talking about why we do taxonomy, but basically facilitates communication, helps you to understand virus evolution. It's nice when you find something new and you can place it exactly where it belongs. And if you find something that doesn't have a place, you create a new one, and helps you to understand the relationships among viruses. Anyway, there's many reasons, but I guess that's enough. Now, you've heard it, millions, right? Literally, Antonio just said 10 million viruses. Can we do it? Yes, we can. Naively, did he say, right? Of course, we're living in a golden age of virus discovery, right, for a few years now. The number of new viruses that keep being reported are just mind-boggling, staggering. I mean, every paper now reports X-fold increase in the number of known viruses, DNA viruses, RNA viruses. And fine, they're being reported, they're being sequenced, they're being deposited in databases. But at some point, they have to be formally classified. They have to be placed into the formal taxonomical scheme, right? They have to be given names. I'm not going to get into names today, but that's part of taxonomy, right? Classification plus nomenclature. So we were not used to deal with this number of taxa, 10 million. But yeah, there is a general consensus among the ICTV that it can be done, okay? And that's mostly what I want to talk about to you today. Why do we think it can be done? And how it could be done? So 10 seconds to tell people that we have a website. So, and by the way, what is the SCTV, right? There is, of course, we're talking about virus taxonomy, but every cellular group of organisms have their own taxonomy organizations, right? Bacteria, fungi, plants, animals, and viruses as well. So we are a voluntary organization, basically comprised of virologists. Everybody's a virologist. I wouldn't call myself a taxonomist. I'm a virologist. I have my own research program. Coincidentally, also on single-stranded DNA viruses, just like Arvind. Arvind, by the way, is a member of the ICTV. He also is part of the committee. Simon is involved in the ICTV as well. Maybe many of you here are involved, and it's a large group of people. I don't know everybody, but yeah, we all have our day jobs, right? But we also do taxonomy, and it's different from other. Well, I just want to show the website. Okay, so here's the address, and there's all these different resources that you find there at the website, and I'll be happy to tell more people about I'll be happy to tell people more about the website later, but that's not the point here. I just want to say that virus taxonomy is unique because the ICTV is responsible for both classification and nomenclature. If anyone here is involved in bacterial taxonomy, for example, there is an international committee on bacterial systematics. That's not exactly the name, but there's an equivalent. They only do nomenclature. They only assign names. In bacterial taxonomy and fungal taxonomy, classification is usually agreed or disagreed upon based on the published literature. More often disagreed upon than agreed. But so virus taxonomy is very different from the other ones because we do both. We do classification and nomenclature. Actually, you would think that this would be the most controversial aspect, but it's not. This is the easy part, really. When I started being involved with ICTV, I had this idea that wow, classification must be really complicated and nomenclature is so easy, right? It's the exact opposite. People have this attachment to names, and whenever you change a name, all hell breaks loose. And that's why I said I'm not going to talk about nomenclature here today, okay? Now, very important, and this is in everybody's minds in the SCTV, we're not a police outfit, okay? We cannot force people to use, to accept the decisions that we make. So we depend on community adoption. So we do a lot of outreach. In a way, that's what I'm doing here, right? And I give these talks a lot to different groups, to different audiences, with all different interests. And it's really difficult to please, I mean, it's impossible to please everyone, right? But we're very mindful that we need to interact, we need to engage with the different communities that use virus taxonomy in their day-to-day lives, okay? Okay. So this is what virus taxonomy looks like today. I put May here, but we work on a yearly cycle. So that's when taxonomy gets updated once a year, usually late April, early May. So that's what we have right now in terms of number of taxa. And that's why the 10,434 from my title comes from. That's the number of virus species that the ICTV recognizes right now. So the 10 million that Antonio just mentioned, well, okay, they're in the database, but they have not been officially classified yet. They have not been given names, and as the taxonomical assignment right now is tentative. At some point, there's a process to make that official. I'm going to mention that process in a minute. And we have a structure, you know, it's typical from other taxonomies, goes from realm down to species. You see the number increase. And you can find this entire taxonomy in the SCTV website. So that's what we have right now, 10,000 species. And it's funny. I know here right now, you're thinking, that's it, 10,000, that's nothing. But sometimes I give this talk to, for example, plant pathologists, and they're like, whoa, 10,000, that's a lot. They think the exact opposite way. So that's, you know, the type of dilemma that we have sometimes. So like I said, it cannot please everybody. Now, this is today, 2022. Now, let me go back a little bit. This is from 2016. Many of you might be familiar with this paper. This field advances so quickly that this is old now, right? I've seen people referring to this paper, oh, that's a classic, right? It's 6 years old. But it is a classic in a way, right? It's a fantastic paper. And when this paper was published, this is a direct quote from the paper. Report the discovery of 1,445 RNA viruses, including some that are sufficiently diverse to comprise new families. And they have here different phylogenetic trees from different families, and everything that's red is something that they were reporting for the first time, the new stuff. So almost 1,500 new RNA viruses in one paper. This was 6 years ago. And those numbers, at that time, we had maybe I don't remember the exact number, but maybe 4,000 virus species classified at the time. So this would be almost 25 to 30% increase in the number of virus species. I'm just showing one paper. There were many published at that time with similar numbers for RNA, DNA viruses, all types of numbers. So the data was starting to come up, and we at the SCTV, we started to get a little bit worried, because how are we going to classify this, right? So as I said, there were many papers similar to this one published at the time, reporting supposedly thousands of new viruses. And it was pretty obvious to us at the SCTV that these numbers would increase, that 1,000 would become 10,000 and 10,000 would become 100,000 and so on. So I hope the clip works. We felt pretty much like this guy here. This is me, and this is what I'm looking at. Okay, this is the wave that's coming our way in terms of new virus species. All right, and we're trying to run away from it. Well, we didn't run away. So okay, so in 2016, the ICTV organized a workshop in Boston at Harvard University, basically to discuss and decide what should we do with that wave, okay? And I like to joke that that was the F cube workshop. What is the F cube workshop? That comes from freeze, flight, or fight. I'm sure you're familiar with the concept, right? What do you do when you're faced with the danger, right? And at the time, we really didn't know. And there were some people in the SCTV that were just basically frozen. They couldn't do anything, and we were about to be swept by the wave. There were those of us who thought we should just flight, run away, like that guy in the clip. And there were some of us who thought we should fight, ride the wave, try to ride the wave, okay? So long story short, fight. We're gonna try to ride the wave, okay? So now that clip was a science fiction movie. This is a real picture, okay? No Photoshop here. This is from Nazaré Beach in Portugal. And there's actually a person here riding this wave. This is the tallest wave ever ridden by us. So we thought we could do that. We thought we could ride a wave, okay? So at that workshop, it was decided that sequences from metagenomics studies that could be reliably identified as viruses could be classified, would be classified. It was an incredible decision. And so to give you an idea, the bacteriologists have not agreed on that yet. They're still debating whether bacteria that cannot be cultivated should be classified. There's a big split in the bacterial community right now. Some people think they should classify them, some people think they should not. We agreed on that in 2016. We published a paper in that regard, explaining that we thought that was important, that we would try to classify them. And that paper has a quote that I'm personally very proud of, okay, that only by accepting that sequences that are generated by metagenomic methods truly represent existing viruses, and by including them in the classification schemes, can we hope to better understand the ecology, history, and impact of the global virome. So from that day on, metagenomic viruses are viruses, basically, no distinction. As long as you can reliably prove that that sequence is a virus, okay? And we don't even refer to them as metagenomic sequences anymore, just call them viruses, end of story, okay? Now, so fine, we decided we can classify them. We want to classify them. How? How are we going to do it? Well, it was interesting that this was mentioned before today. Most people don't realize, but there are more than 1 million species of insects classified by entomologists with species names. There's more than 300,000 species of bugs. I don't remember the exact quote, but I know that quote. It's pretty cool. God has this incredible fondness for bugs, right? It's true. So when you talk about classifying 10 million viruses, it's not that far from that. So if they can do it, we can do it, right? And we're smart people, so we can probably do it. Of course, they've classified 1 million insects over a period of maybe 200 years, right? They had more time than we did, but basically it shows you that it can be done, okay? That's just insects. Insects are a class, right, of animals. So the first thing we did at the SCTV was to establish the framework. And I know you cannot read this, that's not the intention. This is just showing that we have these different ranks. Here's in the ICTV website, more clear, you can see here, realms, kingdoms. This didn't exist before 2020. This was created in 2020, okay? In that 2016 paper that I mentioned, they only talk about families, because at that time, the higher rank we had was order, because we could not establish reliable evolutionary relationships above that level at the time. Once we incorporated the metagenomic data into the viral taxonomical scheme, it was relatively straightforward to come up with realms. And by the way, we call them realms because we didn't want to have the association with the domains of life, okay? So somebody came up with the word realm, which is basically untranslatable into other languages. So if we speak Spanish or Portuguese, we just say dominio, okay? But in English, we have this nice word realm that we can use that distinguishes it from the three domains of life. And so now we have the framework ready, okay, constructed. Now we have to fill it with the different viruses, right, at the different levels. And that's being done step by step. And it's interesting because I just mentioned community acceptance before, and we were kind of anxious when they created all these ranks, whether the community would actually embrace it and use it. And it has been fantastic. It's been used all the time. And you've probably heard here many times during this workshop, people referring to Leonard Viricota or Leonard using the taxon names at the level of kingdom and class and order and phylum, etc. So this has been readily adopted by the community, showing that it was useful and it was necessary. Now, the problem is not, as these days in 2022, the problem is not to assign viruses to the upper rank taxa. That's usually done very easily these days. I'm going to show you a couple of examples of two of these very recent papers that described hundreds of thousands of new viruses, okay? And RNA viruses, but of course there's the same for the other types of viruses. So this one here, as you can see, they were able to assign their viruses very nicely at the level of phylum. Okay, we have 5 phylum of RNA viruses these days. A small number of their sequences were unclassified at the level of phylum. So in this paper, they suggested that these unclassified sequences could maybe be classified into 2 new phyla. So at the level of order, they could basically classify all of their viruses at the level of order. Even at the level of family, they didn't touch anything below the level of family, genera and species, okay? And here's another paper. Simon is a co-author on this one. And the same thing, at the level of phylum, most of the sequences can be reliably classified. Again, some of the sequences would not be classified into 5 phyla. They, I think, also suggested 2 new ones, right? But here they went a little ahead. How they classify their sequences: 7 phyla, 93 classes, so 5 times more classes than they currently exist. As you can see, as you go further into the ranks, the increase, the fold increase is higher, right? And here, although, and I was asking Simon about that just the day before yesterday, see, phylum, class, order, family. This would be equivalent to genera, although they didn't call it like that. But that would be equivalent of genera, and this would be equivalent to species, okay? So 124,000 new species, a 10-fold increase. They didn't dare give them names. Maybe you should try to give them names, right? But yeah, so you can see there was an assignment. And in phage, I know phage people, they usually use this average nucleotide identity of 90%. And that's the type of thing that the SCTV discusses and debates, whether this is correct or not, whether this could be used across all phages, or maybe you should have different species thresholds for different types of phages. These are the type of things that we really go deep sometimes discussing. And oftentimes, this across the board things don't work. It's better if you look at specific groups of viruses and you use specific criteria for that group that may not be the same for that other group, and et cetera, et cetera. But that requires time. So we do revisions all the time. And this could be a first attempt to classify all these new viruses, and eventually subsets of these viruses would be looked in more detail and different criteria would be used for that. That's basically what we do. But as you can see, it can be done. It's already being done, right? And that's why I said we can probably classify them. Now, the SCTV, that's my last topic here. The SCTV has a very detailed decision-making process. This is the boring part, so don't worry, I won't spend too much time here. I just want to show that we have lots of checks and balances, to make parallel with politics. And in this case, it's been working, right? So we have a yearly cycle when taxonomy proposals are submitted, discussed, evaluated, voted for, and if they're approved, eventually they become official taxonomy. Okay, so usually here, this happens in June, this happens in March or April. Okay, and then our databases are updated and the new taxonomy is released. Now, the key thing here is that you need proposals to be prepared and submitted by someone, okay, ideally you. Usually people who describe these viruses, they're not interested in writing taxonomy proposals. So we have study groups in the SCTV comprised of virologists that scout the literature and prepare taxonomy proposals. Now, the problem here is that the way the system is set up right now, these taxonomy proposals, they're basically prepared manually. there is a Word file and there's an Excel file that I have to, it works for a small number of proposals describing a small number of new viruses. No way this will work for 125,000 viruses. So we have to completely change this, and we are aware of that, okay? So how do we do it then? First, the existing procedure, we already agree that. The SCTV already agrees with that. The current procedure does not work, okay? We have to come up with something different. We still have not been able to come up with something different. There was an initial attempt that we called the TPMS. It's been in trial mode since 2018, which is an automated system, computer-based, to write taxonomy proposals using machine learning and everything. But it has many problems, and it really, honestly, I don't see it having much of a future. At some point, we just have to give up on this thing and come up with something new. And I don't have time to go through all the problems, but really, it probably won't be the future. We were also thinking about ending that one-year cycle that I just mentioned and make it a more dynamic process so that taxonomy proposals get submitted and evaluated and voted for at any time, okay? Although we will probably still have to update the databases just once a year. So that's something that we're discussing as well, okay? Then there's the issue of nomenclature. Of course, if you classify 10 million viruses, you have to come up with 10 million names. And believe me, that's a whole other can of worms that I'm not opening today, okay? And before I finish, I just want to say that just last week, I was in a single-stranded DNA meeting with Arvind and other colleagues. And another colleague of ours, who's also a member of the SCTV, just came up to me one night and said, now, do you really think we need to classify all these millions of viruses for there will never be a physical isolate? He was anxious. He was worried about maybe we're getting into painting ourselves into a corner here. Well, I said, well, maybe you don't need to classify all of them at the same time, but I think it would be interesting to have a system to allow them to be classified if we wanted to. That's basically what I'm aiming for. And you know what? 5 years ago, there were all these uncultivated viruses, and many of them has since been cultivated. We've heard some talks today, so it's just a matter of people really studying them in more detail and finding ways of dealing with them. So I still think there is value in having these viruses as part of the taxonomy scheme. And just to finish, as I said, we still don't have a system to replace the current one. There's people working on that in the SCTV, but anyone can help, all right? So I'm leaving my email address here. If you think you can help, if you would like to help, just get involved, okay? As I said, it's voluntary work. You're not gonna get paid for it. You're not gonna be famous. You might get some hate mail sometimes, but I know I'm making an excellent advertising job, right? But yeah, I've been working on the SCTV for 20 years now. It's been very interesting. I like the work. I think it's rewarding. And it's a service to the community, basically. So if anyone wants to get involved, just talk to me, talk to Arvind here. Simon is involved as well. Just get involved, okay? And thank you. Thank you for your attention.